So good morning, everyone. And my name is Henry O'Connell, and I am a consultant psychiatrist working in Portlaoise, Ireland, and associate clinical professor at the University of Limerick, and founding chair of this College of Psychiatrists of Ireland special interest group on evolution and psychiatry. You are very welcome to this, the sixth meeting of our special interest group. We will begin, as usual, with our guest lecture, which will be delivered today by Professor Kevin Mitchell from Trinity College Dublin. And we will have a brief business meeting at the end, so this meeting should be finished by 11:30. You can find lectures from our previous meetings on the College of Psychiatrists of Ireland YouTube channel. To date, we have had lectures from Mike Watts on evolution generally and applications to clinical medicine, by Riyad Abed on evolutionary perspectives relating to eating disorders, Annie Swanepoel on autism and ADHD, Randolph Nessy on why natural selection has not eliminated mental disorders, and Adam Hunt on personality and psychopathology. As always, I would like to thank Helen Murray from the college, Dr. Mara Petrut, Vice Chair of the Special Interest Group, and so many other colleagues who have shown interest and given support to this group since our inception in early 2021. I would like to especially thank my former teacher in physiology at Trinity College Dublin, Dr. Alan Tuffrey, for linking us up with Kevin Mitchell for today's lecture. So on to Kevin. Professor Kevin Mitchell is a graduate of the Genetic Department at Trinity College Dublin and received his PhD from the University of California at Berkeley in 1997, where he studied nervous system development. Following postdoctoral research at Stanford, since 2002, Kevin has been on the faculty at Trinity College Dublin as a Science Foundation Ireland investigator, and now as Associate Professor in Genetics and Neuroscience. Kevin was EMBO Young Investigator and was elected to Fellowship of Trinity College in 2009. He served as Associate Director of Undergraduate Science Education at Trinity from 2016 to 2018 and led a reimagining of the Trinity Science courses. Since 2018, Kevin has been Senior Lecturer and Dean of Undergraduate Studies and has been leading efforts to create a systematic common architecture with the arts, humanities, and social sciences under the Trinity Education Project. His research interests are in understanding the genetic program specifying the wiring of the brain and its relevance to variation in human faculties, especially to psychiatric and neurological disease. He is particularly interested in schizophrenia and autism and variation in perception, including synesthesia. His group has discovered numerous genes involved in specifying neuronal connectivity in the developing brain and shown that mutations in such genes in mice can lead to neurological and behavioral symptoms, modeling aspects of epilepsy, psychosis, and ADHD. Kevin's cross-disciplinary work on synesthesia has helped shape the understanding of the genetic, developmental, and neural basis of this unique perceptual condition. He is also a leading scholar in the genetics of neurodevelopmental disorders, having made numerous theoretical contributions and recently edited a book on the subject. The overarching goal of his work is to help develop and promote a coherent conceptual framework in which to integrate findings from diverse fields, particularly genetics, developmental biology, and neuroscience. This strategy is manifested in Kevin's cross-disciplinary experimental research and scholarship, and also in his teaching, conference organizing, blogging, editing, and other writing. He has developed multiple courses in the emerging integrative field of neurogenetics. He's an active communicator on Twitter and can be found on @WiringTheBrain, and writes a popular blog on the intersection of genetics, development, neuroscience, psychology, and psychiatry with the website www.wiringthebrain.com. His 2008 book, Innate: How the Wiring of Our Brains Shapes Who We Are, develops an integrative conceptual framework in which to consider the origins of variation in human faculties through a novel synthesis of findings from behavioral genetics, developmental neurobiology, neuroscience, and psychology. So we are very honored to have Professor Mitchell today, and the title of his talk is The Whys of Psychiatric Illness. You're very welcome, Kevin.
Thanks very much, and Henry, for the invitation and the kind introduction, and thanks to Alan as well for putting us in touch. It's great to see groups like this that are really interested in the broader sort of biological evolutionary questions around psychiatric illness, and that's what I framed this talk around. The title is a bit odd, "The Whys of Psychiatric Illness," but these are the questions that I'm really interested in here. So the big questions, I think: why do psychiatric disorders exist? Why do they take the forms that they do? Of all the ways you might imagine things might go awry, why do they take those forms? Why do they persist in the population, and why do some people get them while other people don't? And of course, well, to start with, what are psychiatric disorders? Just the categories themselves, I take them to be diagnoses of exclusion. That is, they're based on clusters of behavioral symptoms, where if there's some organic cause that you can identify, well, then that's what they have. But otherwise, they're basically placeholders. So they're not necessarily natural kinds of things in the world that exist unto themselves, as opposed to artificial groupings that we impose on the world. And I'll come back to that point. Clinically, and I should say I'm not a clinician, so I'm obviously preaching to people who know more than I do on this front here, but they're obviously very heterogeneous and dynamic and challenging to treat and diagnose and manage. Now, one of the main sort of characteristics of them is that they're moderately to highly heritable. And the hope is, or the question that I want to explore here, is whether genetics can bring any clarity, first of all, to the clinical pictures and our understanding of what these conditions are, but also to those big why questions that I mentioned at the start. So thinking about common neuropsychiatric disorders, categories like autism and schizophrenia and epilepsy and so on, twin studies and family studies have shown a really, really high heritability. And these heritability numbers are much higher than things like heart disease or diabetes or cardiovascular conditions. So really what that means, the heritability is a technical term. It just means the proportion of variance in who's affected across the population that's attributable to genetic differences. So there's a really big genetic component. Importantly, those same studies have shown there's no effect of a shared family environment. So the goals of genetics as a science generally are to identify risk genes. The point of that is to reveal the underlying biology and hopefully to translate that understanding to the clinic in multiple ways. This can happen just by having genetic diagnoses, but really I think the hope in the field of psychiatric genetics has been to drive personalized medicine and new therapeutics in the way that we've seen in cancer, for example. And I'll come back to at the end to see, how we're doing on that front. But really what I want to talk about is, what does genetics tell us about these questions? And obviously the first area that genetics is most interested in is this question down the bottom: why do some people get these disorders while other people don't? And I'm going to talk particularly about neurodevelopmental disorders, so not the psychiatric disorders that are at the end of the lifespan, neurodegenerative conditions like Alzheimer's, for example. I'm going to focus on the ones that impact at the other end of the life spectrum. They're very prevalent. They range hugely in severity. They impose a huge economic burden. There's lots of unmet clinical needs and social and educational needs. They're largely genetic in origin, but the biology has been very undefined. And traditionally in the field of neurodevelopmental disorders, there's this sort of division between rare disorders that have a known cause, like Fragile X syndrome or Down syndrome or Rett syndrome. And they typically have what's called Mendelian inheritance. So they're caused by one mutation. It can be a point mutation. It can be a deletion of part of a chromosome. It can be an extra chromosome, but it's one sort of mutational event that segregates within families, or that arises de novo. And then you've got this big pool of common idiopathic conditions where they just get a label, like autism or schizophrenia, but the particular causes are not known. what is known, though, from the twin studies, that those are heritable conditions, but their mode of inheritance is generally taken to be complex, which just means it's not just one gene in a family. But complex is a pretty vague term. There's multiple ways in which disorders can be complex. And there's sort of two main models for the genetic architecture of psychiatric diseases. One is the multiple rare variants model, which means that different people have, one mutation that causes them to have whatever the condition is, but those can be in different genes. So you get different genes in different families, and the disorder is genetically heterogeneous. And that's the picture that we see for intellectual disability, for example, and lots of other conditions like retinitis pigmentosa, inherited deafness. That's a fairly typical kind of picture for many conditions. But there's another potential kind of model, which is that actually each case is caused by the inheritance of multiple variants that in order to explain how you would get them coming together, have to be basically common in the population. So these sort of polymorphisms that are just segregating through the population. And the idea is that you can inherit more of them or less of them. And if you happen to inherit enough of them that pushes you over a threshold, then you can pass into this sort of disease state. And that model is called the polygenic model, where there's multiple genetic variants at play in each individual. Now, it turns out there's aspects of both of those models that are actually correct. And people have been looking for rare mutations for a long time, and many of them were found through the traditional methods of cytogenetics. So just actually looking at chromosomes down the microscope, and that's how, Down syndrome and Fragile X syndrome and some of the deletion syndromes like Angelman syndrome were discovered. You can just actually literally look at the chromosomes. The comparative genomic hybridization is a more modern molecular method that does the same thing. It just lets you detect deletions or duplications of bits of chromosomes. That's been very potent. And then more recently, exome sequencing and whole genome sequencing have discovered lots of rare mutations that predispose to these conditions with high risk. So first, just briefly on the deletions and duplications, these can arise in different parts of the genome, excuse me, sometimes in a way that's recurrent, where you get the same duplication arising in different people just because of the way sequences are aligned in the chromosome. And when people started to look for these, what they found was that conditions or deletions like 22q11.2 deletion or 15q13.3 deletion, first of all, could be found at a higher rate in patients, say, with autism than in the general population. But they're also found at a higher rate in patients with schizophrenia or bipolar disorder or epilepsy or intellectual disability. And what you can see if you look across patients who have say 22q11 deletion is that they get really a wide range of expression of those of that genetic insult in different kind of clinical manifestations. And in fact, when people have looked at the general population, what they find is that there's a far lower incidence of these deletions or duplications, but people do have them, and they often don't manifest clinically at all. So there's some really wide sort of spread of effect of these particular mutations, which we see over and over and over again, but they manifest in different ways in different people, which obviously needs explanation. Now, people have gone on to look for just point mutations. So it's just new mutations in the DNA that just arise all the time. They used to be very difficult to find, but with this decreasing cost of sequencing, where we can now sequence the human genome for even less than this and much faster, people have done very large studies. They've started to find mutations in particular genes where, again, what they're finding is there's lots of overlap in these mutations in the risk that they manifest with. So you can have mutations that, like this one, for example, that patients can manifest with schizophrenia or autism or intellectual disability or epilepsy or ADHD or depression or lots of other conditions. So genetically speaking, the risk of these various outcomes seems to be highly shared. And that's true for lots of other mutations as well. And this is an interesting study from a couple of years ago, looking at patients with schizophrenia for rare mutations. And what they have found is a bunch of rare mutations in different genes. And here you see some of the deletions and duplications plotted here as well. And the plot is showing, on the one hand, the odds ratio of the increased risk of having the condition if you inherit one of these mutations. And you can see some of them are massively increased, 50 or 100-fold increased risk if you carry these. And that's plotted against what's called the minor allele frequency. So what this means is that the mutations that have the highest risk are the rarest ones in the population. And you'll see down here some really, really common ones that make individually hardly any contribution to risk. And this is a really important point, because actually what it says is that mutations that predispose to psychiatric illness are being selected against. Right? That's why the more severe ones are less common. And I'll come back to that in a minute, because it's a really important point. So overall, the progress, I think, in the field, we had this in the snapshot in the year 2000, we had this big pool of idiopathic cases where we didn't know what the genetic contributors were. Cytogenetics started to identify some rare syndromes. People often, I think, thought of these as not real. They're not real autism, or they're not real schizophrenia. They were somehow distinct genetic syndromes. But actually, as time has gone on, we have these different modern molecular methods for finding more and more mutations. This idiopathic pool is decreasing all the time. And the pool of patients where we can identify a high-risk mutation is increasing. So I think the idea that these are not real is not the right way of thinking about it. I think instead, what we should think that this idiopathic pool is just an umbrella pool, or an umbrella term for lots of hidden rare genetic conditions. So these common pools may actually be made up of hundreds and hundreds of patients with single gene mutations. Now, that's not sufficient to explain everything that needs explaining, but I think it's largely a correct sort of idea, but we have to make things a bit more complex from there. Let's just, before we do that, take a look at the actual risk genes that have been found. So you can look at all these genes and ask, well, what's characteristic about them? And first of all, they're enriched for brain expression, which is good. That's a good reality check. They're enriched especially for fetal brain expression, which I think re-emphasizes the point that these really are neurodevelopmental disorders. They're enriched for expression in neurons and for functions, neurodevelopmental processes. So all of that is good, and it reinforces, broadly that we know that these are brain disorders of neural development, but they're not. There hasn't been any specific convergence. It's not the case, for example, that schizophrenia genetics has pointed to dopaminergic pathways, and depression genetics has pointed to serotonergic pathways, or any other kind of particular biochemical process. So the convergence maybe that had been hoped for didn't really emerge from the data. So part of the answer to why some people get the conditions and others don't, one of our first why questions, is that they have rare high-risk mutations, and these are under negative selection. So this brings us to our second question: why do the disorders persist? And over the years, there have been various sort of hypotheses or explanations proposed for why you can have these conditions in the population. And people could argue, for example, that really the disorders don't impair fitness. That there can be some benefits to having ADHD or autism in terms of the way that we think about things, or how creatively we can think, and people with schizophrenia, for example. But that's just those are sort of by the by. Evolution doesn't care how creative you are unless what you're creating is children and grandchildren. And these disorders demonstrably do impair fitness, in the sense that people with them are far less likely. First of all, they have much higher mortality, but they have far fewer numbers of children on average. So they're clearly impairing fitness. So that's not the explanation by itself. The other explanations sort of fall under the idea of balancing selection, where you can admit that the people with the conditions, it's bad in evolutionary terms to have them, but maybe the mutations in other carriers actually have some offsetting benefit. So maybe, the siblings of people with, say, schizophrenia, maybe they have some advantages and they have more children, and that offsets the thing. It turns out there's no evidence for that whatsoever. There's no evidence around the mutations that they're subject to balancing selection. Another possibility is maybe the behavior was adapted in ancestral environments. So these are really speculative kind of ideas that maybe, the creativity of being sort of schizotypic would have given some people an advantage in other environments. Again, there's no evidence for those. And in fact, there's no need to think that those, we don't have to appeal to those kinds of explanations. The large effect mutations are selected against. So the condition persists, not because the individual mutations persist, but because new mutations happen all the time. So we can think about the genetics that each of us inherits, all of the variants that we have, and when they have arisen in the past. And if you look at this, the red ones here, each of us carries mutations that are de novo, that they just arose. Our parents don't even carry them. They arose in the generation of sperm or eggs, and they can often be really large effect because natural selection hasn't had a go at them yet. And then we have some recent ones that arose, from recent generations past, and then a whole load of ancient common ones that will have small effect. They have small effect because if they had big effects, they'd have been selected against. So the small effect ones are the only ones that are allowed to become common, basically. So new mutations are happening all the time just because of errors in DNA replication. And we can track those now with whole genome sequencing. And we can see, for example, that, well, first of all, the rate. So each of us has about 70 new mutations not present in our parents, several of which may impact on a particular gene, on any gene. Most of those are from the paternal germline. So the more of them are generated in the cells that generate sperm than in eggs. And actually that number increases with paternal age, which fits with the observation that increasing paternal age is itself a risk factor for things like autism or schizophrenia, or in fact, any Mendelian disorders. So really what we have is this picture of mutation selection balance, where each generation we have new mutations, unfavorable ones are selected against, but new mutations arise again the next generation. And so we don't have to, come up with any other sort of explanation for why these conditions persist, except that new mutations are arising all the time, and the disease can remain prevalent. And that's true for, say, Down syndrome, which, it's practically 100% de novo, but it persists in the population because that event recurs at a certain frequency. So really what that says is that actually the prevalence of the condition depends on what we call the size of the mutational target. So that means how many genes are there that can be mutated that lead to any particular condition. So cystic fibrosis, for example, is rare, right? Because it's only one gene that can be mutated. Whereas these, psychiatric illness is really common. And we have to ask why that is. Why could mutations in so many different genes lead to these particular outcomes? What's the nature of that relationship between the functions of the genes, the proteins that they encode, and the effects of the mutations in those genes? So these emergent psychiatric symptoms. And actually, I'm going to argue there is no relationship between those, which is a weird place to land if you're doing genetics. Okay, so I mentioned that the idea that these idiopathic diagnostic pools are, really umbrella terms for lots of Mendelian conditions is too simplistic, because in fact, the inheritance patterns of those conditions don't look Mendelian, right? I mean, I mentioned, for example, the 22q11 deletion just as one instance, where you can get many, many different manifestations in different carriers. And that's absolutely the norm. That's completely true for all of these mutations. So there must be some other sources of variance that modify somehow the expression of these even these high-risk mutations. And an obvious one is genetic background, but maybe there's also an environmental variance at play. And I'm going to argue that there's also just intrinsic developmental variation as a really important source of differences in how these mutations manifest. So first, for genetic background kind of things, even the most classic Mendelian disorders like cystic fibrosis or Huntington's disease have genetic modifiers in the background that they don't affect whether you get a diagnosis of cystic fibrosis, for example, but they can affect how severe your lung phenotype is, or how susceptible you are to microbial infections, or how severe your gastrointestinal phenotypes are. So if you're looking from the point of view of a phenotype like this, then it's a polygenic kind of a trait, right? There's multiple genes involved in establishing the severity of this phenotype. And I think that's true when we look at the psychiatric phenotypes. That's the level that we're looking at, not the level of whether somebody has, a disease with a name like this. So I think, if we're thinking about genetic architecture, there's not a single answer to this. There's really a spectrum. Some patients may have one really highly penetrant mutation that has a more or less specific phenotypic profile. Those are often sporadic because more severe mutations are basically immediately selected against. So we only see them in de novo cases. You can have other cases where there's actually, it's not just one mutation, it's two mutations or three or four, and they have some interaction between them that gives a more severe phenotype. But you can also have a single rare mutation and just sort of lots of genetic modifiers in the genetic background generally that produce some variability here. So if we're thinking again about the types of genetic variation that each of us have, we have, as I said, maybe 70 de novo mutations. We've got a bunch of rare inherited mutations, but then we've got millions of common inherited genetic variants that are just segregating through the population. And we can look for those, look within these to see, well, do any of these increase risk of the condition? And because they're common and they must have small effects individually, the sample sizes required for these studies are absolutely vast. These are these genome-wide association studies, but they've been done with now hundreds of thousands of people in some cases. And more of these common variants are being detected that do contribute a small risk to a small increased risk of these kinds of conditions individually, but collectively, because there's so many of them, they can make a significant risk. So I don't know if you can see this, but the polygenic score, the overall profile for schizophrenia explains maybe 7 to 8% of total risk. So just to summarize for them, they have tiny individual effects. Collectively, they make a substantial contribution. Again, they're under negative selection. Even though the effects are small, natural selection is really potent at detecting even small effects like that. So it keeps them, not as common as they might otherwise be. And again, like for the rare mutations, the risk is completely shared across diagnostic categories. Well, not completely, but largely shared. This recent paper shows, the overlap in the polygenic risk for, say, bipolar disorder and major depression, or bipolar disorder and schizophrenia is almost completely overlapping here. So again, in terms of thinking about the types of genes, and a lot of them are the same genes as the rare ones, but there are thousands more of them. So they have nervous system expression, that's good. Very, very diverse functions, really no biochemical specificity. And my take on this is that actually the polygenic risk is a very general factor. We shouldn't really try to look within there for particular biochemical pathways, because the effect is not due to individual things acting individually, right? It's a collective kind of burden that I think just reflects general neurodevelopmental robustness. That is how well the developing system can buffer the effects of rare mutations that can arise. So rather than thinking of some cases being caused by polygenic effects and other cases being caused by rare effects, I think they're combined in individuals. So one way to look at that is, to say, well, here we've got some patients, they may have one big mutation or a few, less severe ones, but the effects of those are sort of filtered through the rest of the genome. So through the polygenic burden that they may have. Sex also plays an important part here, with males apparently being less robust, genomically speaking and developmentally speaking, than females, probably because they only have one X chromosome. And so this gives an overall genomic risk that individuals will have, which can explain why, some individuals who inherit a particular mutation go on to develop schizophrenia, whereas another person with a different genomic background, may not show any clinical effects. But that's not the complete story, because while these conditions are highly heritable, they're not completely heritable. Right? There's some other sources of variance that are contributing to who gets the condition and who doesn't. So the risk is inherited, but not the disorder itself. And actually, whether somebody develops the disorder or not, they can pass on the risk as well to their offspring. So there must be some non-genetic sources of variance, and people have thought a lot about that. And typically, the typical conclusion when we say there's a limit to the genetic effects is that people say, well, okay, the rest must be environmental. It's either, if it's not genes, it must be environmental. If it's not nature, it must be nurture. Well, again, from these twin and family studies, we can see, well, nurture seems to make very little contribution to the liability variance. You can see that in adoption studies. You can see that in twin studies and so on. And people have looked, of course, for systematic environmental factors, and there really aren't many that are showing up for risk of these conditions. So we're left still with this question: why aren't monozygotic twins then completely identical? If they share 100% of their DNA sequence, why don't they always manifest with these same conditions? So some other factor that's making them different from each other, not just for these conditions, but generally speaking. So the risk is inherited, and really how it plays out depends on how development plays out. And development is not deterministic. It's intrinsically noisy. There's some random elements to development itself. It's an extraordinarily complex process to wire a human brain. And the genome doesn't actually have enough information in it to specify the outcome. And it doesn't specify the outcome. It specifies the sort of the biochemical rules and processes that allow the brain to self-organize, but it doesn't control every aspect of that beyond specifying those initial rules. So as a result, you don't get the identical result every time you run the program. You don't get the same outcome. And we can see that in the physical faces, say, of twins like these two, who are very similar, but you can see actually not really identical. And you can see it in their brain structures as well. So this is a graph showing correlations in brain, I think it's cortical thickness, between pairs of identical twins or pairs of fraternal twins. And the purpley red bits here are very highly correlated in this measure between identical twins, and less so in fraternal twins, showing it's, it's really, the brain structure is really heritable. But it's interesting that, these areas in blue here are not very well correlated. Lots of bits of brain structure that differ between identical twins, even from a very early age. So by the time their brains are formed, they're already unique, even though they started with the same genetic program. So to get from genotype to phenotype, you have to go through development, and that just adds some variability. It can add variability in quantitative traits, like how tall you are, or the shape of bits of your face, or the shape of bits of your brain. But importantly, brain development has really important non-linearities to it. So events that happen in brain development are contingent on prior events having happened properly. As a result, small differences at one stage can cascade and be amplified into really big differences at the end. Sometimes dichotomous outcomes. And I think, handedness and sexual orientation are good examples of those. There's some anatomical defects like the genesis of the corpus callosum, which displays this kind of all or nothing outcome, where you can inherit a risk of that not forming properly. But even in identical twins, one can have a perfectly good corpus callosum and the other one can have no axons connecting the hemispheres at all. And I think that's true for conditions like schizophrenia or autism. And one way to think about that is this old visual metaphor called Waddington's epigenetic landscape. And epigenetic there just refers to epigenesis, the emergence of the organism through time. And the idea is that if you think of the organism as sort of developing through time like this, then it may have a number of different potential endpoints that it could end up in. But at some particular points, there may be a little bit of noise that pushes development this way in one, say, one twin, whereas another twin who has exactly the same genetic landscape. So this the shape of this is determined by the genome. The shape would look different for different people, but here we've got two people with the same genome where they still end up with different outcomes just because a little bit of noise here, and then the self-organizing processes of brain development channel it one way or another. And to me, that's a really important way of thinking about the way development works generally, but it's really important for these kind of conditions. So we have this overall genomic risk, rare mutations, polygenic risk, sex effects. And then you have this developmental variation on top of that. So you can get some people with no clinical issues. You can have others who have this sort of broad developmental brain dysfunction. But even then, it can still manifest in very different ways in different patients. Okay, so what does genetics tell us about the biology? Well, as I said, we get enrichment for genes involved in neural development, but not really much convergence. And that reinforces the idea for me that actually what we're talking about is some aspect of developmental robustness or its inverse, really a vulnerability. And that's not unusual. Developmental robustness is itself a genetic trait. It's a sort of an overall genetic trait. It reflects the overall integrity of the genomic program. And I like to call that the genomic reserve, just by analogy with cognitive reserve in psychiatry. And basically, the more, the increasing load of genetic variants just across the whole genome will impair the robustness of the developmental program. And so what you can have is that some people with a low mutational load will have a very robust genotype, and they'll be able to buffer the effects of rare mutations. Also in animals where we can see, we can clone identical animals many, many times and look at the outcomes, what you'll see here is a range of outcomes that's quite narrow. Whereas in people with a sensitive genotype, a high mutational load, or in animals, you get a broad range of outcomes, but you would also have a greater sensitivity to the effects of these rare mutations. So that leaves some really crucial questions unanswered. Why do mutations in the same gene lead to different phenotypes? Well, partly we've sort of explained that with genetic background and developmental variation, but the flip question, why do mutations in so many different genes lead to similar phenotypes? We haven't answered that. Right? There's hundreds and hundreds of different mutations encoding proteins with very diverse cellular functions, and yet we end up with these same phenotypes. And why those phenotypes? Why don't we see, any of the range of phenotypes we could imagine for brain dysfunction or cognitive dysfunction that could go in all kinds of different ways? And why do we see clusters of phenotypes along different dimensions that sort of reproducibly occur together, enough for us to say, well, that's what we call schizophrenia, or that's what we call autism? Those questions are not answered by the genetics that we've looked at so far. And in fact, this isn't just a genetic condition. If we look at something like psychosis, we know it can be caused by all kinds of things, right? It can be caused by syphilis or malaria or AIDS. It can be caused by all kinds of drugs that affect dopaminergic signaling, cannabinoids, glutamatergic signaling, serotonergic signaling, all kinds of things. And lots of genetic insults, from deletions to mutations in single genes, encoding all kinds of different proteins. So really, it's a common phenotypic endpoint. And we should stop, I think, looking for some specific or direct link between the particular causes and the outcome, because there is no direct link. These are emergent phenotypes that emerge through development when development is challenged in some way. but it's a property of the perturbed brain. It's not a property of the perturbations that cause it. And this is a weird place to land for people doing genetics, because the whole point of doing genetics is to try and understand the biology in a way that explains the symptoms. And what I'm arguing here is that the biological functions don't explain the symptoms. And that's different for different kinds of conditions. So the point of doing experimental genetics, or in humans or other animals, is that the relationship between genes and functions is somehow parallel to and explains the phenotypes that you see. And that's true for some kinds of things, but it's only true when the functions of the genes are close to the phenotypes that you're looking at. So we take an example of microcephaly, where the brain is just much smaller than it would be, because the neurons and the stem cells in the cerebral cortex, the neural progenitors, haven't divided to create as many neurons as they normally do. Right? So there's a very concrete biological process that underlies this phenotype. And when you do the genetics there, what you find is that the mutations that you find encode proteins that are really pretty directly involved in that process. So they're very proximally related to the phenotype that we look at. Now, when it comes to psychiatric conditions, that's not the case at all. And I like to use this analogy here, where, for example, if we think of microcephaly as like, say, a condition where we know that the turbines are affected, and we want to know, well, how do the turbines get affected? And if you did the genetics of that, right, if you mutated random bits here or damaged random components of this fighter jet, then the ones that would manifest with damaged turbines would be the ones that are in the turbine, right? Because you're looking at a very particular thing. But when we're looking at psychiatric symptoms, we're looking at the highest levels of performance of the whole brain. We're really looking at these this overall functioning of all of the brain and all of the components and how they interact with each other. So we've completely divorced the phenotype that we're looking at from the actual biology of the things that we emerge. In fact, if you damage any of the elements of this fighter jet, you could affect its performance. And I think that's true for psychiatric illness as well. And I think that's why the mutations in the genes that are mutated, their functions really don't have anything to do with cognition or working memory or the other perception or the other things that are affected in these conditions. There aren't genes for autism or schizophrenia. They're not genes for particular cognitive traits, because those functions emerge from the working of the whole brain. So these are genes that impair neural development, where the mutations impair neural development. And then what emerges are pathological states, which now this is I sort of apologize for this because it sounds like a judgmental term. It's an engineering term of art called failure modes of a dynamical system. Right? So any dynamical system that's designed to do something can fail when you start to impair various components of it in sort of qualitatively novel, unanticipated ways. And for me, I think that's how I think of these emergent states. This is another way to think of it, as attractor states in dynamical systems. So the idea is here, you're looking down on a flat plane, and these green contours are like wells in the plane, where, for example, if you had a little marble and it was in this attractor basin, which, let's say, represents the typical outcome of development and a well-functioning brain, then even if you shake it up a bit, it's pretty much it's pretty likely to still come back to where it's supposed to be. That from an evolutionary point of view, right? This is the outcome that evolution has selected on the basis of. However, because the system is so complex, you're going to have some potential maladaptive functional regimes, and if you shake this hard enough, right? If you hit the system hard enough, then it can go out into one of these regimes, and it might be this one, or it might be an alternative regime over here. So the end states, I think, are, in a sense, they are natural kinds, but they reflect the neural system. They don't anymore reflect the genetic system. The insults can be quite varied. Okay, so so just to sum up then, these were the questions that we started with. And I think the answer to why some people get these conditions and others don't informs on, why these conditions persist and so on, and why they exist and take the forms that they do. In the sense that my sort of overall view is that evolution has selected for a complex human brain with incredibly complex cognitive functions. And really, what that means is it has selected for a developmental program to build that human brain with those functionalities. But it has to do that robustly, and it has to be able to buffer all the noise that's inherent in the developmental program and try to channel individuals into a viable outcome. But because it has to do it robustly, ironically, that allows genetic variation to arise and accumulate. Well, it allows it to accumulate. And sometimes that accumulated variation or new mutations is sufficient to push development into one of these alternate trajectories, which can end up in a maladaptive. And I mean that it's not been selected for, but it's very difficult to select against after the fact, qualitatively novel dynamical regime, which manifests as psychiatric illness. So that's my view of the genetics of these conditions and also the evolutionary aspects of them. Thanks very much for your attention. And I'm just going to leave a little plug here for a couple of these books. This is really an academic book about the genetics of neurodevelopmental disorders. This is the one that Henry mentioned in the introduction, which is more, well, let's say more readable, I hope. Thanks very much.
Thank you, Kevin. That was really superb and really enjoyed that. And Kevin, just bearing in mind, I suppose the majority of the audience are clinicians. I know you're not a clinician, but I'm sure you have some idea of the world we inhabit. And I'm just thinking about the challenges we face on a day-to-day basis with our patients in terms of both diagnosis and treatment. And our diagnostic systems are based essentially on symptom checklists, assessment of functionality, and defining syndromes like schizophrenia, bipolar affective disorder. And they're really based on Emil Kraepelin's diagnostic systems from the late 19th century, and I don't think in reality they've advanced that much. So I'm wondering, with these insights from in neurogenetics, do you think we could or should review how we diagnose and classify the mental disorders completely?
It's a great question, and I think I would say yes, but not completely. I think some people, argue that we should move from clinical diagnoses to genetic diagnoses, and I think it's important, if we can, to identify, a high-risk mutation, if it's there. And if it can be found. And I think that's really important, what you can find. And this is happening as, patient groups, especially for these rare mutations, they sort of have sprung up around the world. And that allows clinicians to recognize similarities about, say, patients with 22q deletion syndrome or 3q29 or SCN2A or SHANK1, whatever it is, right? So I think that can be really clinically useful, where you can see, look, there are some aspects to the course of this condition that are associated with this rare mutation that can inform my treatment, my prognosis, my expectations, and certainly can provide, I think, a lot of comfort and help to the patients and their family members in saying, okay, I know what this is, it has a name. Here's what I can expect. Here's other people who suffer from that same condition, the support groups and so on. I think that's super useful. However, at the same time, we have to recognize that any of those rare mutations has quite diverse outcomes. And just because, say a patient comes into the clinic with symptoms of autism and you sequence their genome and say, well, actually, you've got a Fragile X mutation, so you have Fragile X syndrome, you don't have autism. Well, that's not true. They still have the same symptoms that they walked in the door with, right? And so my view is that those two things are complementary to each other, right? If we can get a genetic diagnosis and that's informative and helpful, then great. If we can, but that doesn't mean the clinical aspects are no longer important. And I think, one question is whether the diagnostic categories are really useful or more dimensional symptom sort of things are useful. And I mean, that's a question that you all can answer better than I could. I don't know whether you actually treat autism or you treat anxiety or other aspects. Well, my feeling is you treat the symptoms, not the name, right? So those, I guess, the diagnostic categories prove their usefulness if, for example, they have some indicators for what medications people might respond to, right? So say if you give people a diagnosis of schizophrenia as opposed to bipolar disorder, and that actually has a predictor that they're going to, respond better to antipsychotics than to lithium, then that's a useful.
Yeah. And I agree. I agree 100% with what you're saying. With it, the clinical diagnosis is still kind of the first link. And I guess, thank you for the lecture. It was very interesting. For the main point that it made for me here, I guess, is that so we'll never be able to make our diagnosis solely based on genetics. I guess we kind of knew that with the one single gene non-Mendelian transmission. So the clinical diagnosis is still the main part. The one thing that's missing, I think, is the biology link. So in the clinical diagnosis, not looking only at the symptoms and classifying and diagnosing by classifications, but looking at biology, the brain, the chemistry, what changes can be seen in the brain. And there's not as much, I guess, because I don't know what the reasons are, but it's not as much research done into that and development in that area compared to genetics.
I mean,
and then the biology, and just as you said, for the medication, response to medication, it's a lot better to know the individual metabolism and genetics of every person rather than the clinical classification of symptoms in the prediction of how they will respond to a certain medication.
Yeah, I think so. And one of the goals of doing all this genetics was to get at the underlying biology, and the hope was that by doing that, we could move quickly from identifying a gene, saying there's a mutation in this gene. So the reason that this person is suffering from these symptoms is that this biochemical pathway is affected, and I can add a drug back to that, target it, and tweak it back, right? And it's a kind of a naive.
It's not naive, it's idealistic, I'd say.
Well, I mean, it's founded on the successes, for example, in cancer, where cancer really is a molecular phenotype. I mean, it's a molecular cellular phenotype of biochemical processes in cells, and the mutations that affect those processes, like DNA repair and proliferation and so on, can really be targeted directly to affect tumors. So the idea of sort of personalized medicine and going from genetics to potential drug targets and new therapies wasn't founded on nothing. But my argument here is that actually the symptoms that emerge are not properties of the biochemical functions of the genes that are perturbed, because you've gone through this long trajectory of development, and it's the way that the brain has developed and the states that the brain gets itself into.
But that
which is where the phenotypes are to be explained. And that really means that actually that's a job for neuroscience. It's no longer a job just for genetics. But my hope is that the genetics can inform the neuroscience.
I agree. Can I just add to that? So the prism, the very nice image with the prism turning into a rainbow, very nice with phenotypes. So the biochemistry imprinting of everybody, that's part of the phenotype. That's why we each have, that's imprinted the way the genes develop and the influence of the development along the way. And it does matter. And the older you get, it does matter in the way you respond. And so it's all linked. It's not, that's my point. That the
absolutely. Yeah, I mean, the genotype influences the probability that you'll end up with one phenotype or another.
And that's the whole idea with the disease and this disorder. You can sometimes actually see it in the biochemistry of the body manifesting.
Yeah, sometimes, yeah.
I'm just going to open it up. Is there anyone else who would like to ask a question of Kevin or make any comments? I might just mention then as well, there's a fantastic book called Hidden Valley Road, written by Robert Kolker, on the Galvin family who grew up in the States between 50s and early 70s. And there were 12 children in that family, 10 boys and 2 girls, and 6 of the 10 males had schizophrenia, in inverted commas. Yeah. But he gives a fantastic case description of this. It was a very, very tragic outcome for the family. But he illustrates so well how the diagnosis and our reliance on these categories is oftentimes based on very loose ground, and how the symptoms and the functionality and the profile of these 6 sufferers change so much throughout their lives. And it ties in with your theory relating to development and how and robustness, and all these people presumably had similar genetic profiles. They were siblings, but they all had very severe mental illness manifesting in different ways over extended periods of time. And it shows just how mercurial diagnosis can be, and how changeable diagnosis can be as well. They would have, their diagnoses jumped from schizophrenia to bipolar to schizoaffective to drug-induced psychosis over the years, but they all had very severe illnesses.
Yeah, and that jives well with what I've heard from other clinical colleagues about the nature of diagnosis. It's often a shifting target, and it moves, and I guess again the question is what are the consequences of that? When a diagnosis shifts, does that mean that someone gets a different medication that they wouldn't have gotten otherwise? And for me, the usefulness of the categories comes from whether they actually make predictions that have a benefit. Is it a benefit to have gotten a distinction between schizophrenia and bipolar disorder, clinically speaking?
Yeah.
Or are we just sort of muddling along and trying out various medications in a sort of trial and error way with individual patients over time, and just see what works and what doesn't?
Yeah. Okay. Well, I think we'll wrap it up at that point. And
there is one hand up.
Sorry. Yeah. Can you see that? I'll check the chat.
Rachel Elliott.
Yes.
Yes, I'm not using them. The hospital really covered.
I can't make you out. Sorry.
Could you type it into the chat, please? And there is one more question in the chat.
I can put it in the chat.
Oh yeah, I see the question. I see the question there in the chat already. It's a great, we don't really know, I guess, is the answer to that. The question is, why do some people develop PTSD and others don't in the face of more or less the same traumatic experiences? I think, generally speaking, I think some people are more resilient than others psychologically, but that's not, all I'm doing is paraphrasing the observation there, right? I think some children are more resilient than others to even, neglect and abuse during childhood, where some have very bad outcomes and others seem to come through that, with far less sort of permanent trauma in their own behaviors. So, why is that? You can talk about maybe cognitive, a parallel of cognitive reserve in terms of, emotional reserve. But so for something like PTSD, it's partly genetic, right? So it's a partly genetic condition, but what the genetics is doing presumably is determining or influencing how resilient you are cognitively, emotionally, behaviorally to these kinds of effects. So you're getting, you're getting multiple steps away from any kind of clarity between a genetic effect and an outcome, because now you're adding in this extra complicating factor of an environmental insult, and then the genetics is determining vulnerability or resilience. So it's even more complicated, and I think just frankly we know much less about it. At least I know much less about it. And so, okay, so hopefully that answered that. I mean, it doesn't answer it, but it's a response at least. So the next question, do you think it could be realized in clinical practice for genome sequencing to be used as an adjunct? Are there ethical considerations arising from sequencing patients' genomes to identify risk genes? So yes and yes. I mean, we talked a little bit earlier about how having a genetic diagnosis can be useful. The hope, I guess, would be over, 20 years from now, after we've done all this neuroscience and so on, we might be at a stage where we can say, actually, now we know that, this patient should get this drug and this patient should respond to that drug. That's possible, but for me, it's a long way away. It's obviously useful for things like informing reproductive decisions, for example. So if you, for a family, if they have a child who has a condition, and that you can show that that's caused by a de novo mutation that's not carried by the parents, then that informs reproductive decisions possibly. So I think a lot of the clinical usefulness actually may be in guiding reproductive decisions as much as treating patients themselves. But then that does raise all kinds of ethical considerations, right? And actually, anytime you're dealing with genetic information, it raises considerations like that. I mean, for example, my father did, one of those 23andMe genetic test things, right? And he got his results back, and turns out he carries a rare mutation. Well, it's not that rare here. It's common enough in Ireland, but it predisposes to a serious illness. And he has one copy of it, so he's not affected. But he told me that, and I didn't want to know that, right? So anytime you do genetic information about somebody, you're getting genetic information about all their relatives, and they may or may not want to know that. So, I mean, that's just almost a trivial sort of illustration of the dangers of genetic information and the kinds of uses or misuses that it could be put to, because there's all sorts of issues about insurance, not prejudicing against people because they carry some mutation, for example, and so the ethics of privacy of genomic information, not just for the patient themselves but for their relatives, becomes really, really important. And of course, if you're talking about reproductive decisions and you're saying, well, imagine in pre-implantation genetic testing and in vitro fertilization, for example, we already routinely screen for things like Down syndrome. We can screen for chromosomal abnormalities. You can do whole genome sequencing now of one cell from an embryo, and you can screen for anything, right? So you can screen for more and more mutations, and we'll find more and more mutations and be able to recognize them as increasing risk of these kinds of conditions or other health conditions, but maybe only a bit. Statistically, it's not 100%. So then you're in a kind of a gray area of what we should be allowed to select against in that kind of scenario. And once you're selecting against some things, once the door is open, then it becomes really difficult to make a purely principled ethical argument. And instead, you're in the realm of making pragmatic operational arguments, which is, that's how that proceeds. So anyway, yeah, lots and lots of ethical issues once you're into these genetic domains, I think. And those are just a few of them.
One more question, Kevin, if you're okay.
Yeah.
Today, Rachel Elliott, I often use the analogy of a broken leg with my patients. The problem for the patient is the broken leg, whatever you want to call it. The treatment at the time is pretty similar regardless of etiology, but prognosis recurrence will depend on etiology, i.e. if you jumped and broke it well, don't jump again. If it's due to calcium or tumors, you need a different approach. Yeah. And many thanks for your talk as well.
Yeah. I think that's right. And what it does is it says you're bringing in both the clinical condition, what are the symptoms that the patient is actually suffering from that require treatment or management or support, and then wanting to know, well, what are the causes, right? And of course, sometimes the causes will let you know, should you expect those symptoms to arise again. Sometimes they'll be, in rare cases, there's actually an indication of a medication that you should or should not use, depending on the cause, like in some cases of epilepsy, where some anti-epileptics are contraindicated due to the genetics. Those are very rare. But also, of course, knowing the cause can mean that you also know that you should look out for other things in these patients, because that group of patients also has heart disease, or they also have early onset Alzheimer's, as in Down syndrome, for example. So My feeling is, I guess, the more information, the better. And it's not the case that the genetic diagnoses trump the clinical diagnoses, or vice versa. I see them as complementary. And I guess the goal would be to work both of those into clinical practice in ways that, you know, we test are informative and useful for the outcomes for patients.
I think Rachel sums it up well when she says then that your talk illustrates that a unique focus, such as only on genes or diagnosis, is not helpful as well.
Yeah, no, I think that's absolutely the picture that emerges. Yeah, and you know, it's a frustrating one. It would be nice if there were simple, direct answers, but the biology is telling us there aren't. And I guess I get a little frustrated when some people in the field of psychiatric genetics are trying to make parallels that are, you know, that are too direct, too linear, and too simple, when their own results are showing no, it's really, really complex, and we just have to get to grips with that. That's the message. Unfortunately, it's messy.
Yes. Okay, well, thank you again, Kevin. Really enjoyed the talk, and we're going to have it on our YouTube channel, so I'm sure everyone would be watching back over it, because there was a lot of information to digest. Yeah. But I think it really rang true with me. I think it really helps me in terms of how I conceptualize mental illness as a clinician. And so thank you so much for your time today.
Great stuff. Well, thank you all for your attention, and thanks for the great questions as well.